All right, so today we're beginning photosynthesis. This is probably more what you were thinking in line of when you were thinking about, I'm going to take biology, I'll learn about plants and animals. So up till now, I've completely confused you with lots of chemistry. And today's no exception to that. There's still more chemistry, because today we're learning about how we're going to form macromolecules from the radiant energy emitted by the sun. So it's all about converting energy from one form to another. What's the study of converting energy from one form to another called? Thermodynamics. In this case, what we're talking about today is converting energy from one useful form, one unusable form, to a useful form. Now you see this all the time. What's this big globe thing? The sun, which is also a star, which surprises people, I know. But the sun is a star. It's emitting light. It's emitting visible light and radiation. That light is going to strike objects on Earth and allow those objects on Earth to do work, to do things, because the organisms can convert that energy into usable energy, into free energy. We need to power our biosphere. The biosphere is wherever we have found life from the bottom to wherever we find life at the top. Wherever we've looked, we've found life of some form or another. We can dig and dig and dig and dig. And the farther down we dig, the more, well, not the more, but we keep finding organisms. Cryptoendoliths and such, bacteria living deep under the Earth's core. We can go up and up and up. And we find life right up until we hit space. So the biosphere is anywhere you've got life. This biosphere needs energy to go on. It needs energy to survive. And one way that we think about that is to think about food. Now, you've got two big types of organisms, heterotrophs and autotrophs. Heterotrophs are like you and me and cows. They're organisms that take in organic molecules from the environment. We eat to get organic molecules, and we break those substrates down. We oxidize those substrates. We steal energetic electrons from them. Autotrophs, on the other hand, organisms that can create their own organic molecules from inorganic sources. So, going through this real quick, is this a heterotroph or an autotroph? Heterotroph, because it's eating the zebra. How about these plants growing here? Autotroph, they're making their own food. And then this zebra? Heterotroph. I know, that's, some people get thrown off by the zebra. So we can get sources of energy from lots and lots of different sources. We can create macromolecules using the energy from the sun, which is probably our most important source. Lots of plants use the sun in order to create macromolecules, but it's not the only source. Deep at the bottom of the ocean, you have these geothermal vents producing a whole lot of ions that can be broken down by the microorganisms living there. Things that have never seen the sun before can use that energy to create food to sustain an ecosystem. If you ever get a chance to watch a documentary about or read about the organisms living at the bottom of the sea, 2 miles underwater, there are thriving communities around these vents because they're using the energy produced by the Earth's heat and the ions produced from these plumes to create life. But at the surface, we got a much more abundant source of energy in the sun. Photoautotrophs, so not just autotrophs that take in inorganic molecules and create organic ones, but photoautotrophs are going to take the sun's light and convert it into usable energy, usable macromolecules. So you guys know these are plants, right? You'll see these leaves. Leaves are what color? Green, and typically they're going to be flat. Why are they flat? They're flat in order to increase the amount of surface area they have. Do you guys know what this is? This one in the bottom left corner? Yeah, it's a type of cactus. Aloe vera. You guys heard? What's aloe vera good for? Sunburns. People can take it and break it off and rub it on their skin. Although I can't tell you to do that because apparently that's a misdemeanor. because it's a controlled substance. You're effectively, I know, crazy talk, right? Aloe is on this controlled substance list because you're using it to do as medication without a medical license. Isn't that weird? Which is why if you go to Lowe's and you look at the tag for it, it doesn't say good for burns. It says great decorative plant. Does that make sense? Well, that's what I'm saying. It's actually classified in the same way marijuana is, which is hilarious. Do you guys know what this is? It's a type of flower, yeah. What kind? It's not tulips.
It's not a monogamy, but it's similar.
It's similar. Yeah. No, it's not. Well, no. How about this? It's very important to this region, or historically has been. It looks sort of like a bouquet of flowers. But you usually don't see that part. It's tobacco. So there you go. Tobacco can be pretty. The thing is, though, once it gets to this point, you've gone beyond the useful, the use of the leaves. The sugar's been put to the wrong place at that point. So today we're going through photosynthesis. Photo meaning light, synthesis meaning creation. So using light to create things. You're going to bond together carbon dioxide and water. So CO2 plus H2O is going to yield oxygen, O2, which is the toxic byproduct produced by plants, and C6H12O6, glucose. How you guys taken chemistry before? You remember stoichiometry? Wasn't that super fun? Real quick, we can get this whole equation. Now, that fairly simple equation is going to be broken down into two phases. The light-independent reactions and the light-dependent reactions. The light-dependent reactions are going to, of course, require light. These are known as the photosystems. The light-independent reactions are also known as the Calvin cycle or the Calvin-Benson cycle. They're going to happen whether or not light is present. So before we talk about photosynthesis, we need to set the stage. Where's this whole thing happening? And we know what's happening in the leaf, right? The leaf is green and flat. Now the leaf itself can be divided up into segments. The middle of the leaf, if you were to take the leaf and sort of rip it along its axis, you'd end up in what's known as the mesophyll. Meso meaning middle. So photosynthesis happens in the leaf. What part of the leaf? The mesophyll. The middle of the leaf. Now, to get into the leaf, to go from the outside leaf to the inside, you got to go through these little mouth structures called stoma. Lots of them are called. A single one is the stoma. Many of them are stomata. Water can also escape. So while these mouths are open, you're getting carbon dioxide in. You're also losing water. So a plant will ideally have these closed more often than not. But they can open up to allow carbon dioxide in. So photosynthesis happens in the leaf in the mesophyll. In the mesophyll, you have cells. In the cells, what structure, what organelle is where photosynthesis occurs? The chloroplast. So in the cells, you got the chloroplast. The chloroplast itself has structures within it. Inside the chloroplast, you've got these little disc-like structures. Those disc-like structures are called thylakoids. Now, these disc-like structures all have a membrane. What's that membrane made of? It's all membranes made of in the cell. Start with the P. Pardon? No, the plasma is the phospholipids. Remember phospholipids? And whenever you think membrane, think phospholipids. So in these membranes, you have lots of proteins embedded. So the proteins of the thylakoid, the proteins embedded in the membrane of the thylakoid, inside the chloroplast, inside the cells, inside the mesophyll, inside the leaf, on the plant, are where photosynthesis occurs. Do you remember a song is when you were younger, the Rattling Bog? No, okay, never mind then. There's a song called the Rattling Bog. It's all about, was it like a twig on a branch on a limb on a tree and a hole in the Rattling Bog? Okay, thank you for one person understood that reference. Same idea. In this case, photosynthesis happens in the membrane of the thylakoid and the chloroplast and the cells and the mesophyll of the leaf on the plant. All right, so we set the stage. We know where this is happening. First thing we're going to talk about are the light reactions. What is this molecule right up here in the top corner? Pardon? Well, that's a phosphate, yeah. Triphosphate. What kind of triphosphate? You're right. It is triphosphate because there are 3 phosphates. A little bit more. Just a little bit more. That's all you need. Adenosine triphosphate. Exactly right. So this is adenosine triphosphate that's easily recognized because it has 3 phosphates. What if it had 2 phosphates? Diphosphate. All right, what's this molecule? Nope. That molecule, what is it? You got this, guys. Mary Jane. Do you know what that is? No? Rashedra? No, wait. Is that Mr. Bell? No, okay. Chris, how about you? It is not diphosphate. Tara, I got hope in you. What is this molecule right here? You don't know? Tanya? Hannah? Ishmael? Jamal, do you know what this molecule is right here on the screen? Because you're minimally observant. Anybody know? This is one molecule. Really, guys? Jamal, are you sure you don't know what this is? Do you want me to tell you guys what it is? Yeah, it's NADPH. Yeah. How could I tell it was NADPH? Because it's written right there. Yeah, that's it. So there we go. Oh, yeah, there you go, Terry. Totally obstructed view. It wasn't your fault. All right, so ATP and NADPH are both what? No, they're not both photosynthesis. Energy intermediaries. Remember, they are going to be what move energy around the cell. You can build them using oxidation reduction reactions. Endergonic and exergonic reactions. All that stuff we talked about last time. So here we've got our chloroplast. We're going to have our light reactions occur inside the chloroplast, in the membrane of the thylakoid. You'll notice here you got your thylakoids. Your thylakoids are all these discs that are stacked up. When they're stacked up, they're called a granum. Just in case I ever throw that at you. So you've got your thylakoids and a stack of thylakoids called a granum. It's sort of like if you've ever had Mentos, the thick pack of Mentos. The individual Mentos would be the thylakoid. The stack of them's the granum. Now, you guys remember the first law of thermodynamics? Right. Energy is neither created nor destroyed, only transferred from one form to another. And that's what we're looking at here. We're going to convert the sun's light from radiant energy, from radiation, from visible light, to usable chemical energy. And we're going to do that by exciting electrons. Now, to understand this, you have to understand that light is energy. We are not going to get into some fairly deep physics about the nature of light. Light both acts as a particle and a wave, and we'll just say that's magic and it works. Here's the deal. You could think of light being passed in little packets of energy called photons. Those photons are moving along a wave. The wider the wavelength, that is, the wider the space between this peak of the wave and that peak of the wave, is going to mean it has less energy. Have you ever been to the ocean when it's calm and you have gentle waves lapping on the shore? And there's space in between the waves, time in between the waves. It takes a few minutes for, not a few minutes, a few seconds for one wave to crash, and then another wave to crash, and then another wave to crash. It has low energy on those days. If you increase the energy, let's say a hurricane, more energy in the ocean, the waves are moving much faster. When that happens, one, the peaks are higher, but also it's crash after crash after crash, to the point where all these waves are interacting with each other. This blue wavelength, you'll notice the peaks are much closer. It's got a shorter wavelength, more energy. So longer wavelengths have less energy, smaller wavelengths have more energy. Red, very long wavelength. Blue, short wavelength. So blue has more energy than red. Questions on that? You can pass that energy onto an object. When we're looking at what's called the visible light spectrum, it's called visible light spectrum because it's the spectrum of light that we can see. You've got your reds on one end, and you've got your purples on the other, your violets. It's almost like a rainbow. Our eyes are specifically adapted and evolved to see this spectrum of light, but there is other radiation out there. You might have radiation on this far end over here, below red, otherwise known as infrared. You have microwaves down here, radio waves down here. Do radio waves hurt you? No, radio waves don't hurt you. This energy, it doesn't have much energy. AM versus FM. AM have even longer wavelengths than FM. They travel farther with less energy. On the other side, you've got closer and closer and closer wavelengths. Up here you have ultraviolet radiation, X-rays, gamma rays. The farther down you get, the tighter those wavelengths get. Is this dangerous? Yeah, this is the nasty stuff. Ultraviolet radiation, that stuff the sun can hit you with. Ultraviolet radiation can lead to mutations, breakdown of your DNA. X-rays, you know those are dangerous. Whenever you go to the dentist or to the doctor to get X-rayed, they put a big lead shield on you and then leave the room, which is always very comforting, isn't it? They're like, stay right here, I'm going to do a dental X-ray. I'll be in the other room behind this giant lead shield, but you'll be fine. Gamma rays. When you get to gamma rays, you either have instant death or you become the Incredible Hulk, and there's not really much in between. Gamma ray bursts could wipe out half the planet if the star was to emit a gamma ray burst and it hit the planet. The people on that side of the planet would be dead before they realized what hit them. And it's not like walls can protect you from it. So that's fun. But what we see is this visible radiation. Red, low energy. Violet, high energy. When light comes through and it strikes an object, it can do one of three things. First thing it can do is just go right through that object. It's transmitted through it. It's like a window. If you look out the window, light can come through. Any sort of light, if we look out this window, you can see through it. Light is being transmitted through that window. It's not stopping, it's not bending, it's not distorting. Another thing to do is light might be reflected. If light comes down and strikes an object, it can be reflected off. But it may be that only certain wavelengths of light are reflected at any given point. My shirt, what color is my shirt today? Not trying to trick you. It is blue, that's right. My shirt's blue. It is reflecting blue wavelengths of light. It's absorbing the other wavelengths of light. So this apple, This color, what color is the apple? Red. Thus it is reflecting what color? Red. All of the other wavelengths of light are being absorbed by it. In this case, blue and green. Coming in, striking the apple, and being absorbed. Now, when they're absorbed, that object is absorbing their energy. Does that make sense? Think about the color black. Black absorbs all wavelengths of light. White reflects all wavelengths of light. In the summer, do you prefer to wear white or black? White, because it keeps you cooler. The light is striking you and being reflected. You are not absorbing any of the energy. If you wore black in summer, what happens? You get hot because the light strikes it and doesn't reflect. Your clothes are then absorbing all of the energy, heating up, kinetic energy. My hair gets very hot in summer because I have dark hair. Some of you guys have dark hair too, right? When you touch your hair in the summer, it's like, ow, it literally hurts. For you blonde-headed people or light-haired people, you don't have that issue. You'll never understand our pain. So light can either be transmitted through an object, so you can see through it, it's transparent. It can be reflected off an object, so you can see the color that's the wavelength reflected, or it can be absorbed by that object, transferring its energy to it. Now, plants are going to use only visible light for energy. That visible light spectrum will strike a plant, and they will absorb only those wavelengths. Some of the things they reflect, some of it get absorbed. What color light is reflected from plants? They're reflecting green. They are not absorbing in the green wavelength. They're only absorbing in the blue wavelength and the red wavelength. So what would happen, looking at this week's experiment, if you were to put a plant under green light? It would die because it's reflecting all that light. There's no energy that it's absorbing. Inside of plants, you have these things called pigments. Pigments are molecules that are going to absorb energy of a certain wavelength. You might have cyan pigments. Cyan absorbs red, but reflects blue and green, so you get that sort of light color. Let's see, how can I put this? The pigments that are in plants will absorb specific wavelengths while reflecting others. When light strikes a pigment, this molecule that absorbs energy, what happens, back to the original point of the course, the original big components of the course, when light strikes any object, or when any object molecule gains energy, what happens when a molecule gains energy, an atom gains energy? Right, well, sort of the electron cloud expands, right? The electron cloud gets bigger. As visible light strikes an object, it absorbs that energy, the electron cloud expands. And now you have an excited electron. And that excited electron can do work. It might do work for that molecule, or it might be transferred to another molecule. The pigments in plants are called chlorophyll A, chlorophyll B, and carotenoids. These pigments give rise to the colors of plants, and they also, more importantly, are going to absorb energy for these plants. Chlorophyll B and chlorophyll A are going to both absorb in the blue spectrum, and they will also absorb in the red spectrum. So what you're seeing here is the amount of absorption. You'll see a lot in blue and a lot in red. Where do you not see a lot in? Green. Green, there's no absorption. If there's no absorption, what's happening? It's being what? Reflected. So what color are you going to see when you have chlorophyll A and B in a plant? Blue is absorbed and red is absorbed, so what's being reflected? Green. Thus, plants are green. There are other pigments, though. These pigments are typically going to be found in lower quantities in plants than chlorophyll A and chlorophyll B. They're called carotenoids. These are less abundant in plants, but they're still there. They're going to absorb in the blue and green spectrum. So if they're absorbing blue and green, what are they reflecting? Red and orange. That's where you get your carrots and you get your oranges and all these other colors. So I refer to them as carotenoids. You may hear them referred to as carotenoids, and that might help you remember it. The problem is I learned plant physiology from a Canadian and from a British man, which means that my pronunciation of some of these terms is off from what Americans pronounce them as. Does that make sense? So I'm sorry if I screw you up. But these carotenoids, or as the Americans call them, carotenoids, are going to be oranges and reds because they're absorbing blues and greens. This is what gives rise to the colors of fall leaves. Fall leaves are going to be reds and yellows, same colors as the carotenoids, because what happens is the quality of the chlorophyll A and B decreases. Thus, you're able to see the carotenoids, which exist in lower quantities. For instance, my shirt, what color is it again? Blue. My shirt has threads in it, though, that are white. If I was to remove all of the blue threads, the only threads you would see left are white, and my shirt would begin to look white. Does that make sense? It'd be very threadbare. But you see what's predominant. Usually with leaves, you see chlorophyll A and chlorophyll B. But as those levels decrease in the plant, you end up seeing the carotenoids expressed, which gives you the colors of fall. So we're going to use that energy from that. So light's going to come in, strike the plant, we get some energy from it. We're going to use the energy from those, from the photons striking a pigment to excite the electron. That electron will then be able to do work for us in the photosystems. The photosystems are proteins and pigments working together in the membrane of the thylakoid in order to create an electrochemical gradient that powers ATP synthase. I'm going to go through the photosystems very quickly. Then I'm going to go through it step by step. Then I'm going to go through it again. The photosystems are complex, and I'll tell you, you will have an essay on them. So to begin with, I'll give you a second to finish taking notes. For the next, say, 2 minutes, I'm going to ask that you just pay attention and don't try and keep up taking notes. Just listen to what I say. Then we're going to go through, and you can take notes as we're going through it in depth, okay? Then I'm going to say it again. But I really want you to just sort of try to get the whole picture before we concentrate on the individual steps. So step 1, you've got your thylakoid membrane. Now remember, the chloroplast is double membrane bound, right? Here's your outer level of the chloroplast, here's your thylakoid. So what's going to happen is you have a pigment in the plant. That pigment can absorb energy. There's the sun. The sun produces energy. That energy is in the form of photons. The photons come down and strike the pigment. When it strikes the pigment, an electron is excited. An electron gets energy. So when the electron gets energy, its electron cloud expands. It can do work now. Now, what's going to happen is right next to that pigment, there's another molecule, another protein. And that protein is going to steal the electron because it's more electronegative than the original pigment. It steals the electron and now can do some work. And the work that it does is it pushes hydrogen across a barrier. Next, there's another set of molecules, another way of transporting hydrogen. It's more electronegative than the first. It's going to use the electron to push hydrogen across. That electron's going to jump again, pushing hydrogen across. We're building an electrochemical gradient until eventually that electron runs out of energy. Its electron cloud has decreased in size back to where it was originally. Now you're going to hit the final electron acceptor. This final electron acceptor, I believe, is called QB, And it is so highly electronegative. It is the most electronegative molecule we're going to look at. It will steal an electron from oxygen. Now, you guys know oxygen's super electronegative. It steals an electron from oxygen in water and converts it into O2. So we've taken this electron, we've used up all the energy of the electron to push hydrogen across the gradient to create an electrochemical gradient. And in the process have converted water into oxygen. We're out of energy. So what do we need? More energy. Where do we get more energy from? The sun. Got another pigment. More energy comes down, strikes that pigment, excites an electron. That electron can do work. In this case, the work it's going to do is it's going to convert NAD, NADP into NADH. So you have removed some hydrogen from down there, further increasing the electrochemical gradient. And here that gets converted. And now you're running out of energy again. All that's left is in this section you've got ATP synthase. Now we talked before about chemiosmosis. ATP synthase is going to take this electrochemical gradient. It's going to allow the hydrogen to flow through it. And in the process, it's going to convert ADP into ATP, which is an energy intermediary. This section here, this first part, is called PS2, or photosystem 2. This part's photosystem 1. Photosystem 2 comes before photosystem 1. They did that just to mess with you guys. Seriously, the reason they're named this way is because photosystem 1, this ATP synthase, was discovered first. Photosystem 2 was discovered second. That's the big picture. If you can explain that, you're set. We're going to go through step by step. So step 1. We have this light harvesting complex. This pigment is going to be in the chlorophyll, in the thylakoid membrane. You're going to have to absorb light from the sun. So the sun's energy comes down and strikes that pigment. In this case, it's absorbing red, it's absorbing blue, it's reflecting green. This is what gives plants that green look to them. So sunlight comes down and it strikes a pigment. That excites an electron. So that's the first step. Light strikes a pigment. Step 2. That energy from the light is absorbed by the electrons, giving them more energy. They can do work now. They're highly excited. They're moving quickly. So we've got an excited electron. Yay, I can do work. That electron's ready to do stuff now. From there, that electron's going to jump from one molecule to the next. The first molecule is more electronegative than the pigment. It jumps to that first molecule. It uses up some of its energy to push hydrogen across the barrier. It's going to jump again to the next molecule that's even more electronegative than the first, to a third. And then finally, it's going to jump to a fourth molecule called QB. QB is extremely electronegative, more so than just about any other molecule we'll ever study. So after you've excited the electron, it's going to jump from one molecule to the next, embedded in the membrane. Each time it jumps from one molecule to the next, it pushes hydrogen across the barrier. Finally, it hits that QB. And when it hits QB, QB is so highly electronegative that it's going to steal an electron from water to form O2. So now we've created the water in our big equation for photosynthesis. I'm sorry, we've created oxygen in the big equation from photosynthesis. So now we have our our electrochemical gradient, super highly powered. We've gotten all this hydrogen in between the thylakoid membrane and the outer membrane. We're ready to move. Problem is, now we're out of energy. We have all of these hydrogen on one side, but we're totally out of energy. So that's the end of PS2, photosystem 2. The net result of photosystem 2 is we have an electrochemical gradient produced, and we've converted water into oxygen. Now we're going to move into photosystem 1. In photosystem 1, more energy comes from the sun. It's going to strike another light harvesting complex. When it does that, when it strikes this other light harvesting complex, that excites an electron. That electron can now do work. That excited electrons pass from one molecule, from that electron carrier molecule from this light harvesting complex to NADP. This is a protein. A protein is just a big molecule, though. It's a macromolecule. So NADP is going to be converted into NADPH. So now that that NADP is lost to electrons, it's got a hydrogen from the outside. Now you've got an even stronger electrochemical gradient. You're able to do even more work. You've got a huge buildup of hydrogen ions. So the big takeaway message is that you are pushing a whole bunch of hydrogen ions outside of the thylakoid space in order to create this massive electrochemical gradient. That massive electrochemical gradient will then pass through ATP synthase. All of those hydrogen ions go through one at a time. And as they go through, they're going to take an ADP into phosphate and crush them together to very efficiently make a whole lot of ATP. That process is called chemiosmosis. We talked about it. Is it last lecture or the lecture before? Lecture before. So here we're using chemiosmosis to make ATP in a very efficient way. So the light reactions have used this electrochemical gradient to make ATP. We have usable cellular energy now. The big difference between photosystem 2 and photosystem 1 is that both of them are going to help produce the electrochemical gradient, but photosystem 2 produces oxygen. Now that oxygen is actually a fairly toxic waste product produced by plants. Oxygen is so toxic because it's an oxidizing agent. It will steal electrons. You've seen what oxygen does to iron. It breaks it down into rust. Oxygen will steal electrons from things. Early in Earth's life history, this oxygen event, when we first got plants, led to enormous amounts of mass extinctions. Yes, you and I need oxygen right now. But that's only because we evolved in an oxygen-rich atmosphere. If you were to surround a plant with oxygen, they would die. It is a toxic waste product to plants. Think of it as it's plant flatulence. It's like locking yourself in an elevator after you've had a whole bunch of beans. You don't want to be there with yourself. So now, from these light reactions, we've produced a whole lot of energy. Adenosine triphosphate, energy intermediaries, we've produced NADPH. And we've produced this toxic waste product of O2. So those are the light reactions, light dependent. They are called such because they depend upon the sun. Questions so far? Do you guys understand? So far we've covered this conversion of water into oxygen. Huh. Sorry, I just noticed that this summary of photosynthesis is wrong. It says 6H2O yields 6H2O. It doesn't. There should be a 6O2 over there. You're converting water into oxygen. We've done that. And we've built up a whole bunch of ATP and NADPH. Now remember, those stoma are what's going to allow carbon dioxide in and water out. We haven't yet talked about carbon dioxide, but we're going to need our carbon dioxide in order to produce glucose, that C6 part of glucose. So the light-dependent reactions produced a ton of ATP and a ton of NADPH. But we have not yet produced glucose. The next three steps are going to be what we use to make glucose. It's called the Calvin-Benson cycle, otherwise known as the light-independent reactions. There are three steps to the Calvin-Benson. I'll write them down for you. 3 steps to the Calvin-Benson cycle. 1, carbon fixation. 2 is known as reduction, or you might call it carbohydrate production. And 3 is the regeneration of RUBP. So we're going to have fixation, reduction, regeneration. Or fixation, carbohydrate production, regeneration. Those are the big steps. These 3 steps are going to allow you to take carbon dioxide molecules and form sugar. Now, if that doesn't sound like magic, I don't know what does, because you're literally taking gases from the atmosphere and turning it into sugar. Now we need to build molecules. We're going to build glucose, which has lots and lots and lots of bonds in it. In order to build a bond, what do you need? Energy. Well, we don't have the sun anymore. The sun's gone down. These are light independent. So where do we get the energy for this? Say it again? Yeah, you stored up some from those light reactions. You made a lot of ATP. We can use that energy to build this massive molecule with all of these bonds to store all of that energy. For every 6 carbon dioxide that come into the plant, you're going to use 18 ATP and 12 NADPH. You're going to hydrolyze them. What does it mean to hydrolyze? To break it up using what? Water, hydrolysis. And each time you break that up, each time you break these ATP or NADPH, what's released? Energy. Now you are oxidizing them. If you are oxidizing these things, ATP and NADPH, what are you doing to them? You're forcing them to lose electrons. You're stealing electrons. You're taking energetic electrons from these energy intermediaries and using them to power your own reactions. Build bonds. So we've got 3 big phases: carbon fixation, reduction, and regeneration. We'll go through each of them. The first step is carbon fixation. You're taking carbon dioxide, CO2, and binding it to this molecule called ribulose bisphosphate. So ribulose, R, ulose, U, bis, B, phosphate, P. Carbon CO2 gets attached to RUBP to form a molecule called 3PG. You don't need to know about 3PG. But you do need to understand that you're taking carbon dioxide and attaching it to RUBP. That process is not easy. In order to make that process happen faster, what do you need? An enzyme. So you're going to use an enzyme to attach CO2 to RUBP. The enzyme that you use is called Rubisco. And I always think of it as a cookie. I don't know why. I think of Nabisco. You know, the little wafer cookie. Anyways. Rubisco. Ribulose, bisphosphate, carboxylase, oxygenase. But everybody just calls it Rubisco. Rubisco is probably the most abundant protein on the planet. There is more Rubisco out there than any other protein in any other organism. Because it's in all of the land plants. It's in all of the aquatic plants. Anything that has photosynthesis has Rubisco. And the whole job of Rubisco is to take carbon dioxide and RUBP and force them together. Actually, Rubisco can do two things. It can take carbon dioxide out of the air, and then combine it with RUBP. So Rubisco grabs carbon dioxide, crashes it onto RUBP, and now you have 3PG. It's an enzyme-mediated reaction. So what happens to this at high temperatures? It happens to in a high pH. It doesn't work. Why? Denatures. You lose the shape. You lose the form. You lose the function. If you add inhibitors to this, it slows down. If you increase the amount of Rubisco, it speeds up. It follows all the same rules all the other enzymes we looked at follow. So we take this carbon dioxide out of the air. We crash it onto RUBP with Rubisco to form 3PG. The other steps, and I'm not going to go through each and every stupid individual step, is we're going to take that 3PG and build sugar from it. Now basically what happens when you build sugar is you're going to take that 3PG and you're going to break it apart and put it back together in a new way. Just like you would do with a Lego set. You break it apart, recover some energy, put it back together. In the process, you've destroyed your ribulose bisphosphate, your RUBP. But you've made sugar. So during step 1, you take carbon dioxide and carbon dioxide and RUBP and you put them together. In step 2, you break that structure apart and build glucose, but in the process, you've broken apart all your ribulose bisphosphate, your RUBP. So then your final step, you have to rebuild your RUBP, and this is where most of the energy goes. You're going to take a bunch of your ATP and use it to rebuild the RUBP, because without more RUBP, the process couldn't continue. You need a cycle. So step 1, you use your RUBP and your carbon dioxide. Step 2, you break them and build sugar. So step 3 has to be rebuild the components you need, rebuild the RUBP, regenerate it. So there we go. Fixation, carbohydrate production, regenerate RUBP. You can see carbon dioxide, and I'm not making you memorize this. You take carbon dioxide in, and it binds to RUBP to form that 3PG molecule. That 3PG is then going to go on to become sugar. But in the process, there's the sugar coming off. You've broken the RUBP, so you have to recover it. So you have ribulose phosphate. You need ribulose bisphosphate, so you have to add another phosphate to it. And now it's a cycle. Here, you're taking that carbon dioxide and attaching them together with Rubisco. That's that enzymatic process, creating 3PG. In the process, that 3PG is going to be broken apart, put back together to form glucose. All you have left is ribulose phosphate. You have to add a phosphate back to it to make ribulose bisphosphate, and you can enter the cycle again. 3 steps. Fixation, reduction, regeneration. Fix the carbon dioxide. That means attach it to RUBP. Reduce it or create your carbohydrate, and then recover or regenerate your RUBP. There we go. So that's photosynthesis. Probably way more about photosynthesis than you ever wanted to know. Downside is it can screw up. And the reason it can screw up is because it is an enzymatically mediated process. It needs enzymes. So anything that will affect enzymes will affect this. So the net result is we've taken the energy from the light-dependent reactions, photosystem 2, photosystem 1, and we've used them to create a molecule of glucose. So now we have glucose present. It works for plants. They can make their own food. From there, that glucose can move to the mitochondria. And what happens in the mitochondria? Energy gets made. You have aerobic cellular respiration. The glucose is broken down. Problem is, it doesn't always work perfectly. You might end up with a process called photorespiration. In this case, you're going to accidentally, instead of binding carbon dioxide, you bind oxygen. That's not going to work out so well, because you need carbon dioxide to build sugar. Without carbon dioxide, your wheels are going to spin, but you're not going to produce anything. Now, this happens in oxygen-rich areas. Again, it's a toxic byproduct for plants, oxygen. So if you have too much oxygen present, you might accidentally grab it instead of carbon dioxide. Also, if it's too hot. If it's hot out, the shape of the rubisco changes, and you end up accidentally, as you try to bind carbon dioxide, that rubisco will accidentally bind oxygen instead. It's all because rubisco is stupid. Rubisco is not very smart. It doesn't always bind to carbon dioxide. Rubisco will sometimes accidentally bind to oxygen, especially if it's hot, especially if there's a little bit of a pH imbalance, especially if there's too much oxygen present. So we need to account for that. And to do that, we have different types of photosynthesis. C3 photosynthesis, C4 photosynthesis, and CAM photosynthesis. I've been meaning to, and I have to go back and write a section on CAM plants. But the idea is C3 and C4 both start, C3 starts out with a 3-carbon molecule, C4 with a 4-carbon molecule. CAM plants are going to be like your cacti. They have a totally different way of undergoing photosynthesis. But every organism, whether they're in the desert, down by a creek, in the ocean, has to be adapted to its own environment. So we need these different mechanisms of photosynthesis. C3 photosynthesis is what we've all learned about. We just learned C3 totally, you know, was it 3GP, or I'm sorry, 3PG is C3 photosynthesis. That's everything you've learned up till now. You start off with a 3-carbon molecule. See, 1, 2, 3 carbons. So the first molecule in C3 photosynthesis has 3 carbon. That's why it's called 3C, because C stands for carbon. You got lots of different examples. Oak trees, soybeans, strawberries, potatoes, wheat. Any of these plants that require a lot of water are going to be C3 photosynthesis. Typically, they exist in cool, moist environments. Anything down by a creek is going to be a C3 plant. Usually, if they're in the shade of another tree, C3. They're cool areas. They're moist. Has lots of water. In C3 photosynthesis, the photosynthesis occurs throughout the leaf. Any of the cells in the mesoville, you have C3 photosynthesis going on. Aside from the chloroplast, there's no specialized anatomy required. You just need chloroplast and you're good. The problem with C3 photosynthesis is it's got very poor water use efficiency. The plant has to keep its stoma open longer. It's not very good at converting carbon dioxide into sugar. It accidentally brings in oxygen. It has photorespiration occur all the time. So because of that, it has to keep its stoma open. It has to keep its little mouths open. And when the mouths are open, it also loses water. This is not a problem in an area where you have a lot of water. Sort of like you guys go to a buffet. What's a good a buffet restaurant around here? Do you guys know any place you can get? Golden Corral. All right, you go to Golden Corral. I've gone to Golden Corral. I know this place. Okay, so go to Golden Corral. My kids love going to Golden Corral because they can eat whatever they want. If you go to Golden Corral, you get a plate of food and you don't like it. What do you do with it? You throw it away. Yeah, just leave it there. Somebody will take it away. Who cares? There's always more food. Does that make sense? You're looking at me like that's crazy talk. Really? Do you eat everything on your plate all the time at Golden Corral? Yeah, really? Even the bad stuff?
I mean, you get to pick what you eat, so why would you get the bad thing?
Because your parents are there and they're judging you.
Okay.
You don't care? So, most people, unlike Jamal, or let's say you go to CC's. Do you eat the crust at CC's? Do you like crust? If you don't like crust, do you need to eat the crust when you go to CC's? No, you don't have to. You can leave it aside because there's always more. Does that make sense? There's an abundance of food present. So you don't have to eat everything if you don't want to because by God, you're an adult and you can do that. These plants are the same thing. They don't care about how much water they've got. They live next to a creek. There's always more water. They'll just suck that up. They're fine. See, an issue is Rubisco is pulling double duty. Rubisco is not just grabbing carbon dioxide. It's not just attaching RUBP to carbon dioxide. It also has to grab the carbon dioxide. And Rubisco is dumb. It screws up. It grabs the wrong thing in too much heat. So to account for that, it's got to keep its stoma open longer. It's going to have more carbon dioxide coming in. It's losing more water, but that's okay because there's plenty of water where that came from. Those temperatures really impact the reaction rates of Rubisco. It really, really slows it down because it forces photorespiration to occur. Now, the way I always think about photorespiration is you can think of it as like an overworked clerk. Somebody who's processing paperwork. You guys have probably dealt with financial aid at some point in your life, and you probably came away not liking that experience. What happens is you grab the wrong paperwork accidentally. That stops the whole process. You fill out Form 1B instead of 37A, and suddenly you've screwed up, you've got no money, so you don't get to go to school this semester. Does that make sense? One little change, the clerk grabbing the wrong form, or in this case, the Rubisco grabbing oxygen instead of carbon dioxide, is going to throw off the whole process. Now, when you think about that, you guys ever worked in a place that didn't have air conditioning? I have. It's not fun. It's actually what made me. I worked as an electrician for a while, and then I realized I don't like heat, and that didn't work out for that profession. When you get hotter, you make more mistakes. If it's 90 degrees in here when you're taking your exam, I guarantee your exam scores are going to be lower than if it's this temperature. So higher temperatures, these enzymes are bound to make mistakes because there's slight changes in the overall form of the enzyme. You can also think of it as controller mapping. How many of you guys play some games on a console? Okay, if you play console games, the worst, and the rest of you just have to deal with this. The worst thing you can run into when you're playing a game on a console is somebody remapping your controller. I know, it's the worst, right? You're sitting there like, all right, I have to, I don't know, whatever you're playing, you got to swing a sword, say, and somebody remaps it to a different value. Suddenly you're ducking instead, all the time. I played a game called The Witcher. If you ever played, some of you may never have heard of this game. Anyways, in this game, I swear to you, I walked into a wall for like 20 minutes because I couldn't figure out how to do things. The controller made no sense. Photorespiration is like controller remapping. Photorespiration is where you think you're doing something. You think you're undergoing photosynthesis, but you've grabbed the wrong molecule. and instead are doing something totally different. You thought you were doing something useful, and it turns out it's totally wrong. You're not getting anywhere. You're wasting a lot of time. You're wasting the batteries in your controller. You're wasting your time and energy. And maybe eventually you'll get to the end of a level because you'll curse at the screen long enough. But in the end, you don't account for anything. You don't accomplish anything. Photorespiration doesn't give you anything good. It just makes you spin your wheels, and it makes the plants use energy. It's not good. So we want to avoid photorespiration. We want to avoid these problems. Now, C4 photosynthesis helps you avoid these problems. Instead of using a 3-carbon molecule, it's got this 4-carbon molecule. Not terribly important, but it tells you why it's called C4. Examples of this are corn, Bermuda grass, sugarcane. Have you seen this grass growing in Danville? You probably have. It's like this big, long strip of grass. It's like this long wire that will go across the street and implant itself wherever it can. I assume you've seen this. It's growing in my yard. Somebody in Danville, this is not endemic to the region. This is not part of our region. Somebody thought it was a good idea to bring this because it was for their turf. So this grows really, really well in bright, sunny areas. It doesn't grow well in the shade. It doesn't grow well in the cool weather. It doesn't grow well by a creek. Other plants outcompete it. But in bright, sunny, dry areas, that grows really well. Corn, same thing. Lots and lots of sunlight, lots of heat. Sugar. These are C4 plants. They typically occur in hot, dry regions. Think the savanna. Think the edge of the desert. Now in C4 photosynthesis, you have new structures forming. Photosynthesis takes place in the inner cells of the mesophyll. So if this is your leaf, you have epidermal cells on the outside. You've got a stoma. Stoma lets in carbon dioxide, lets out water. And you've got cells surrounding the stoma. And then next to those cells, you have the inner cells of the mesophyll. Photosynthesis is going to occur here. It does not occur here. These are the outer cells of the mesophyll. There's no photosynthesis happening there. Only in these inner cells. That arrangement of cells, this structure here, these circles are called Kranz. What are they called? Kranz structures. The Kranz anatomy. Kranz is a German word for wreath. So they're sort of wreath-shaped. So there's no photosynthesis on these inner cells here. Lots of photosynthesis, on the outer cells here. Lots of photosynthesis on the inner cells. That Kranz anatomy is going to be key. Because ultimately what you're going to do is you're going to move carbon dioxide from the stoma to those cells. This is very, very efficient when it comes to water use. Because you have enzymes in here called PEP carboxylase. PEP. Write that down. It's going to come up on your exam. PEP will bind carbon dioxide. It will take carbon dioxide out of the atmosphere. But here's the deal. It will only take carbon dioxide. It will never, ever, ever, ever take oxygen. So in these cells of the outer mesophyll, there's PEP. It grabs carbon dioxide from the stoma and carries it across the cell to these inner cells. So PEP will only grab carbon dioxide. The inner cells have rubisco. So what ends up happening is PEP, which can only bind to carbon dioxide, carries it across the cell and drops it off at the rubisco. And the rubisco then takes that CO2 and combines it with RUBP to form the 4-carbon molecule. It's very, very efficient because now the stoma don't have to stay open. You never, ever, ever, ever have photorespiration occurring. Nobody ever remaps your controller. The clerk always gets the right paperwork because PEP is only ever allowed to take carbon dioxide. It will never mistake it for oxygen. So as far as the overworked clerk goes, now instead of them just grabbing papers that they think they need and they can make mistakes, you have one other person there whose whole job is to put the right paper in front of them. And that's the only paper they've got. They can only give them the correct paperwork. It makes it go efficiently no matter what the temperature is. So C3 plants, you got that first organic molecule is a 3-carbon. C4, it's a 4-carbon. In C3 plants, it happens in cool, moist environments. C4, it's in hot environments. C3, the photosynthesis occurs throughout the whole leaf. C4, it occurs in the mesophyll. C3, you don't need anything aside chloroplasts. C4, you need that Kranz anatomy in PEP carboxylase. C3, rubisco can pull carbon dioxide out of the atmosphere and attach it to RUBP. In C4, PEP carboxylase is what pulls it out of the atmosphere, while rubisco's whole job is to combine it with RUBP. So it's a lot. There are a few ways that I highly recommend studying for this. Draw the photosystems over and over and over again. Find somebody and teach it to them. If you haven't invested in a whiteboard yet, something you can draw on and erase and draw on and erase, do so. It will help you immeasurably. Find somebody who doesn't know biology who's willing to spend some time with you and talk to them about this stuff. Over and over again. They will get tired of you, but you will learn it. Does that make sense? And truthfully, at some point, they're going to try and learn it so that they can show that they're smarter than you. Because that's what friends do. I know this is tricky stuff, but I have faith in you. I will tell you straight up that one of your essay questions on your exam will be to describe the light reactions of photosynthesis. That means go into detail about photosystem 2 and photosystem 1. I won't accept just a picture of it. You have to explain the picture. If it helps you to draw a picture and then explain it, that's fine, but you have to explain it. A shorter essay is going to be to explain the Calvin-Benson cycle. Explain carbon fixation, reduction, and regeneration of RUBP. Don't just list them, tell me what happens. So this exam that's coming up is going to cover a whole bunch of things. I have faith in you. I know you can do it. You've made it this far. You have questions?